So I think there's probably, one very important thing with metabolomics is that metabolomics could potentially point us to pathways. Even if the molecules would not be the molecules that are causal to the disease, you could still see how they are affected and so show us the way that things are happening. Okay, that's very relevant. Now, you could also find molecules through methodologies that come from epidemiology that actually try to find causality. And I'll give the example of, say, lactosylceramide. We have published a paper on GWAS, Proceedings of the National Academy of Sciences, just last year, in which we found, we collect these genes that are associated with Alzheimer, say ABCA7, that's one of them, and then ABCA7 is very associated with these molecules that are called lactosylceramides, okay, which are precursors for a number of molecules that do recognition in the cell membranes. And so we found causality that actually lactosylceramide could be causal to the Alzheimer's disease, using these Mendelian randomization approaches that our group uses in epidemiology. And so I see then these two ways. One of them is just to look at molecules that, although not causal, you can see that is affecting that pathway, and so there's probably some relation. So you could look for what's responsible for that, and at the same time look for molecules that are actually causal, and and kind of try to separate the ones that are more relevant for that and the ones that are not.